Hi, I'm Alex Aravanis, Chief Technology Officer at Illumina. Oncology is the disease most commonly associated with genomics. In fact, cancer is often referred to as a disease of the genome, since the DNA of cancer cells is profoundly altered with mutations, tearing chromosomes apart and creating changes to the epigenome. The scientific community has generated an amazing amount of data, identified countless variations of significance, and are systematically characterizing the genomic landscape of cancer. Today, we're going to discuss how this growing amount of data on cancer genomes is creating precision medicine for cancer care. Our panelists are driving this revolution and working at the cutting edge of research and technology, each with their own brand of innovation towards the common goal of improving the diagnosis and treatment of cancer. Joining us today is Elli, Papaemmanuil, Assistant Professor of the Computational Oncology Service at the Memorial Sloan Kettering Cancer Center, where part of her multifaceted program integrates experimental and computational approaches with the aim of developing clinical decision support tools in oncology. Also with us is Joel Dudley, Chief Science Officer at Tempus Labs, a technology company that advances precision medicine through the practical application of artificial intelligence in healthcare. Previous to Tempus, Dr. Dudley was Associate Professor of Genetics and Genomics Sciences and Founding Director of the Institute for Next Generation Healthcare at the Icahn School of Medicine at Mount Sinai. And finally, we have Christina Curtis, Associate Professor and Endowed Faculty Scholar at Stanford, where she leads the Cancer Computational and Systems Biology Group and co-directs the Molecular Tumor Board at the Stanford Cancer Institute. Christina's research combines experimental and computational methods to improve the diagnosis and treatment of cancer. Let's start with, what is the most exciting science that is happening in this field?
Well, there's so much exciting science. It's really hard to pick one. I almost feel like I'll have to pick one at random, from my head. But I think some of the really interesting science that's happening is how do we connect across multimodal data sets? I mean, it's obviously the business we're in, and we're sort of spoiled here with the group of folks that we've assembled in this discussion, because I think we have people who are very excited and pushing the envelope of sort of multimodal data and sort of systems medicine approaches to cancer. But the power of having multiple layers of data, such as RNA, histopathology imaging, DNA, medical records, and so on, collected on the same individual, is that you can start to draw really powerful insights across these data modalities, right? So we sort of started cancer precision medicine with targeted DNA panels, and now we've just sort of blown past that very limited view of cancer. And once you've assembled all these different modalities, you can do really powerful things across these modalities. For example, there's sort of a very robust science around what we call histogenomics, right? Which is how do we use artificial intelligence, deep learning methods to actually extract molecular insights just from H&E, whole slide imaging, which is already collected as the normal course of care.
And we definitely share the vision and passion for making the genomics technology more widely available to researchers and to patients and their providers. Christina, how about you?
Sure, yeah. I mean, certainly echo that this is really prime time for being able to integrate these heterogeneous data sets and really to draw on real world evidence as well. And I think that there's a lot of enthusiasm, certainly some challenges ahead in how we can do this. But I think, using the patient as the model, the individual as the model, and being able to garner information from both the EMR, but also more routine testing. And I think there's some exciting examples that are coming out of that. There's clearly a lot of hard work ahead, but thinking about how we can move forward with in silico trials and so forth. And then I would just extend that to say, maybe it's one more, but phenotype, right? I mean, we can measure phenotypes at this point, and how we move from genotypic space to phenotype and really link across clinical parameters is going to be very transformative, and we're sort of just at the beginning of that, but exciting frontier ahead.
I'd have to agree with both Christina and Joel that we have an ever-expanding array of pretty robust tools now in our toolkit that allow us to reliably and automatically extract signals at scale across tissue types or phenotypes. So the technological challenge has been addressed. We have a very good understanding of the algorithmic approaches that are required to convert those signals into the necessary information. And then the next challenge is to try and understand how these link together and how we can interpret them. And that in turn has brought in a real revolution into the integration of data scientists into everyday laboratories. And it brings together very powerful expertise from both the medical, biological field, as well as the more data analytic, statistical learning approaches, coming together to combine our respective skills and try and produce some really impactful new insights, as well as steps forward in our field.
So I found that in all of your responses, implicit was that as sequencing data and the ability to generate sequencing data comes down in cost, it enables you to do science in new ways, to ask different questions. It'd be great to hear maybe some examples of how that's changed over the last few years and how you see that changing, right? So if sequencing cost becomes 10 times lower in the coming years, how does that change how you'll approach asking these questions, doing science, how it might change the translation into the clinic?
It's clear that genomics will become sort of a part of our everyday life when it's cost effective enough. And I think in terms of the research questions that can be addressed, we've already seen just sort of collating the wealth of data that's out there. I mean, there's hurdles in annotation and curation, but that we can start to ask some pretty powerful, clinically relevant questions with the data we have in hand. And that's only going to expand. I mean, there's questions that are currently off the table because we don't have maybe enough data, but that's changing. And, I'd say an example of this that I think personally has been really exciting is how we can leverage this type of data to identify biomarkers that might allow us to improve stratification, and both in terms of prognosis, but also treatment response, and tailoring therapy so that it's more effective up front, but also that we can spare patients unnecessary interventions. Chemo, in some instances, I think is a really important goal of precision medicine. So, just very enthusiastic about having more data to work with and move those questions forward.
Christina, I know you're doing a lot of work in breast cancer and translating some of these insights into clinical trials. Could you give us an example of that, just to really make it concrete, how the technology is being used to think about how to treat breast cancer patients and how to think about how to treat them long term?
Building on discoveries that were enabled through data science approaches with large clinical cohorts, we've been able to identify new subgroups of disease. Some of these subgroups really define groups that are at really high risk, for example, of relapse. And they also define key genomic drivers that are potentially therapeutically targetable, both with existing therapies and new therapies in development. And so this has really led us to think carefully about how we can better stratify. It's led to some new investigator-initiated and federally funded clinical trials to bring these therapies forward. And that actually requires stratification up front. It requires that we deploy those biomarkers in a rapid time frame.
Elli, I know that you're pioneering a lot of work in CHIP, or clonal hematopoiesis of indeterminate potential, and what these mutations that individuals develop over their lifetime might mean for their chances of getting disease, or what it means when they unfortunately are diagnosed with the disease. Maybe you could share some thoughts about how genomics is helping you answer those questions.
Yeah, so first of all, in the field of CHIP, which is clonal hematopoiesis, which represents the detection of pre-malignant clones in the blood of otherwise healthy individuals, has very much been enabled by the availability of high-scale, deep genomic data that has been made publicly available through prior exome studies. So that led to the characterization that quite a substantial number of us carries those pre-leukemic clones in our blood. And most importantly, that we can use high-depth genome technologies to detect these clones reliably. So that immediately opens up the opportunities of, okay, now we can identify who has these clones, but what can we do about this? So by integrating these rich data sets at population scale that are linked to good phenotype outcome as well as exposure data, not surprisingly, we're learning that as everything in cancer, we're dealing with a complex and multifactorial problem. The effect of mutations changes on the base of the cell lineage, the allelic state, which other mutations are present, and what exposures are surrounding those cells that may kind of have interactions with the actual mutations to alter the clonal trajectories of those cells. Understanding some of those effects would have been impossible if we didn't have both the technologies and the data sizes that we have started to look at. And that helps us shape the questions in a way that can inform future study design in a way that's powered to address those very questions.
Well, looking forward, as the cost of sequencing continues to decline, and thinking of new applications there, I would like to put a emphasis on collecting longitudinal data, dynamic data, right? So I think at Tempus we generate a whole transcriptome RNA-seq on every single sample that comes in the door, which I love, and it's because it's a great dynamic representation of the tumor, but it's still only a snapshot in time, right? So I think the next frontier is really getting dense molecular data longitudinally on a patient. Human biology is complex and dynamic and context dependent. It changes over time. We really are largely still collecting snapshots of these individual patients. And obviously we have liquid biopsy efforts underway, but a lot of them are still, it's like the early days of tumor sequencing, right? We have personalized MRD, but it's really targeted to a small number of genes. Hopefully we'll get to whole genome MRD, and then we'll get whole genome MRD with DNA, with epigenetics, with transcriptome, with single cells, and to develop these sort of digital twin, type technologies and virtual trials like that. That's the data set we need to build going forward. How do we get this into the clinic? And we still have a long way to go here. This is, one of the primary missions of Tempus is to how do we deploy precision medicine at scale? And we always say that we're building all the boring plumbing that nobody wants to build to really deliver this at scale. A lot of precision medicine work is, boutique, happening in isolated academic medical centers, doing great work within those academic medical centers, but having very difficult times, to scale that beyond a particular medical center. So that's really one of our missions is to connect everybody up, to enable that scale and to enable others onto our platform, to scale up the innovations that they're developing. I always like to say that Tempus really isn't a testing company. Tempus is really trying to be the Amazon Web Services of precision medicine. One of the major limitations is the capabilities of electronic health record systems that are the primary interface of the clinician, right? So these systems were optimized for billing and recording transactions such as lab orders. They're not smart. So how do you develop intelligent diagnostics, whether they be purely digital, such as EHR, predictors, or molecular, or combinations thereof? So Epic and Cerner are the 800-pound gorillas, but then we work with many community hospitals. So we've basically built a whole system and API, if you will, to take in everything from paper records, which we still get a lot of today, that we have to scan in OCR and structure, everything to pulling clinical data from a Cerner cloud or Epic cloud instance, on the more sophisticated end. So we still have a long way to go, and it's a tremendous amount of work to sort of create a unified interface to that very important clinical data that we need to contextualize every patient record.
You touched on some really important points there. I think one was often what we refer as data engineering. So in addition to data science, you need a lot of infrastructure, right? As the data sets grow, as the number of patients, as the different modes of data, it's really not an easy task to assemble this, to process the data. And it gets harder the more richness and the more we add to that. Bioinformatics, artificial intelligence, or machine learning. Would love to hear your thoughts on what are the differences between these, right? Because I think they are different. And what are the implications for those and how you use them in the work you do? Maybe we'll start with you, Elli.
We're increasingly learning how to develop this plumbing, right? That enables to take a patient's biopsy and return deep phenotyping data for that patient's biopsy to the oncologist within what is now highly clinically relevant timeframes. And then the next big challenge is how do we bridge the gap from that report and reading that report to an interpretation? And I see bioinformatics more as the discipline that ensures that we have high quality signal extraction from the data, so even though there are gray areas right now. It's such a broad field because you have pure bioinformatics, can we extract the signal, and then you have more quantitative statistical learning applications on that data. So the first challenge that we've been dealing over the first years is get the highest quality, most reliable signals out of that data. And then where AI or machine learning or deep learning, whichever way you want to name it, comes in is to say, how do we bring all of that information together? How does that help us classify patients within a group that we understand, then translate to clinical decisions and help us also predict outcomes or optimize treatment decisions as to something that might not benefit a patient or something that will benefit a patient. And bringing those disciplines together are critical to help us bridge that gap of interpretation. And it's just going to be a path of continuous incremental wins across kind of the space.
I really like your framework of kind of, and appreciate that there's a lot of complexity and these applications are broad and techniques, but, bioinformatics is helping us get those signals from the noise and machine learning really interpreting those to ultimately create insights and hopefully actionable ones for patients. Yeah. How do you think about this, Christina?
Yeah, no, I think Elli hit it right on the head. Essentially, we have the algorithms, we have many of the methods needed to ingest these different data types. And I think there's still some important gains that will be made by properly integrating these data, and that's really where the ML or the machine learning approaches come into play. And I think there's a lot of activity that's taking place around this now that we have sort of coherent data sets where we can begin to learn from. And so, not only integrating across these multimodal measurements, but the dynamic component, right? As Joel alluded to, what we really want is to be measuring patients throughout the course of their disease. And we know that treatment, implies heavy selective pressures in some instances, so we need to repeat those measurements. And then how do we assimilate what we saw before with, what's now taking place? So we view it very much in the same way that really now a lot of the fun begins because we have these coherent data sets, at least in some instances, and we need to ingest more and more data and build that out, particularly with the clinical component, and really learn from these different sort of orthogonal readouts. So I think it's a very exciting time. I can see there have been some early wins, and I think if we want to bring this really to every patient, though, we need to become better at detecting these sparse signals, these rarer events, the ones that occur in concert. It's seldom a single event. And so I think that's where a lot of the attention will be applied going forward. And fortunately, we've, as a community, have been learning about some of these tools. And it's very much prime time for us to build on this infrastructure.
These technologies and this know-how, as Joel said, should not be confined at just certain institutions that can gather the collective expertise that's required, but they should really be democratized and available to every patient, no matter where they live or where they're being treated. And also the availability of technologies that can reliably deliver. You have benchtop technologies now that can turn around genome profiling data within hours, very much enables those and software tools that can be cloud-based, that can link through the data from those technologies, can very much democratize the availability and the delivery of this type of solutions to as many patients as possible. And then comes what Joel was saying, how do we integrate all that data and ensure that we learn from them and derive solutions that can go back to the patient. So we'll create this feedback loop between the technology, the solution, the patient, and then back to learn and develop new solutions where we haven't been able to tackle before. It's a fascinating time.
So Joel, we were hearing about some of the ways in which machine learning is creating more insights from these data, and also making it easier to automate and bring these insights together, so that it's not only a handful of experts in the world, but can be kind of more routine to understand how best to use the data. So would love your thoughts on this.
I think one of the things I've been saying lately is that biology is dead. There's only computational biology, right? So we used to distinguish that, but I think when we say biology now, it should just be implied that it's computational biology at this point, because it's basically, obviously, statistical learning and machine learning approaches have infiltrated almost every aspect of biology, even the wet lab space, obviously. But when I think about AI, which I always like to joke is that statistical learning with good marketing. But it's but I really think about it as not just the methodologies that are being developed, but it's fun to dive into those because they're really interesting and really fun to look at the latest deep learning methods and things like that. But I think it's the confluence of those methodological developments plus the technological developments, which I think Elli and Christina had touched on. It's not that these methods exist. It's how are they made accessible, and how are they even to operate at scale? At Tempus, again, we're very focused on scale more than anything. We generate about 2 petabytes of data a month, which is a tremendous amount of data. So you can have fairly straightforward statistical learning methods. You don't even have to go to deep learning. You can look at something like regularized regression or something and things like that. But if you try to run those methods at the scale of data, I think our total data set is 40 petabytes at this point. So something like that. Even running basic statistical methods at that scale is very difficult. Again, I guess I keep going back to the boring plumbing that needs to be built to really make all this stuff operate at scale, but it's super important because it's not highly non-trivial to scale up these machine learning methods to vast, very, very large data sets.
I'd say one thing that's important also to emphasize, and I'm certainly learning this on a day-to-day basis, is that there's not going to be one size fits all in terms of those models. We find in our work that we may have developed a model that achieves the highest predictive power in one disease area, and then you move to the most adjacent disease area in the WHO classification, and that's not the optimal model that works there. So, and that's where joining disease expertise and joining forces with clinical care teams and building communities that speak the same language so that they can come together and use these exciting new toolkits to answer those fundamental questions and challenges that have been the clinical care teams have been dealing at the front line for years is very, very important. Highlighting the need for the communities to work together and implement those solutions to tackle each disease area's problem specifically.
Yeah, I think that's a great point, especially what Christina mentioned this earlier, which is we're in the early stages of phenotypes. And I think that's worth highlighting again, because when we talk about machine learning, what does machine learning need? It needs data and it needs labels, right? So, and we have a long way to go. I think Elli made a great point, is that not only do we have trouble translating between labels, but we also have to now start to figure out, are our labels correct? Like, are we actually categorizing these diseases correctly? And if not, that could have a huge effect on the performance of those models.
Yeah.
Absolutely. And I think that comes back to sort of what we've all been speaking to, which is that we need to be far more dynamic in how we learn from these data, how we integrate, and we, that's only feasible when all of this architecture is in place, when the data have been ingested and curated in a way, and we have enough of them to actually start to iterate in that manner. And I think, this is where I see that there's going to continue to be really tremendous advances. We're really just at the cusp right now where I think we have enough data for certain indications to start to do that kind of, to start to deploy some of these learnings and have really very powerful and predictive models. And in other places, we haven't even gone there. So there's really a huge frontier ahead. But we do know what tools to deploy, and I think that's helpful, right? We're not inventing this necessarily from scratch. Many of these machine learning techniques have been around, as Joel said. It's not that we actually always need something super sophisticated if the hypothesis and the data are appropriate for that question, right?
So maybe it'd be fun to continue on this theme. So what do you think some of the both opportunities and challenges are in aggregating these data? Because we have more and more institutions generating larger data sets. Would love your thoughts on that, because we're all going to succeed, faster and better and help more people. if we can do that. Can imagine a variety of practical challenges, but how do you see us overcoming those?
I mean, I can jump in since we're in the business of doing this. There's a huge number of challenges that need to be overcome. Still, first of all, it's very expensive to aggregate this data, of course, and it's one thing to aggregate data, and it's one thing to sort of make it live data and make it hot data. We've always had things like NCBI Geo and dbGaP and these sort of repositories to aggregate data, but anyone who's ever worked with those repositories knows that's not instantaneous live data. It's actually quite cumbersome, to use those resources. So it's one thing to aggregate. It's another thing to make it the aggregate data accessible and live and queryable and usable.
So I think about this in two ways, and it's a really important point. There is enterprises like Tempus or molecular pathology departments within academic hospitals or hospitals have a de facto infrastructure that warrants an aggregation of data just by the necessity to produce clinical grade sequencing data. So that comes with an underlying infrastructure. And that creates at least a level one for having this in a place that can be organized in a similar way, phenotyped in a similar way, and then that enables dissemination where that is desired. And then I think that the vast body of data that's being generated across research labs around the world, like you mentioned, biology is now computational biology. If there is an experimental model of disease biology, that will invariably result in genomic RNA-seq and increasingly multimodal single-cell RNA-seq data. There are precious samples that have been stored over years with significant investment in biospecimen banks that are converted into data across labs and research institutions around the world. And what I see that these are often very project-oriented, PhD, postdoc, or even within the pharmaceutical industry. They're very structured and organized around projects, and that often results in data silos. And I often wonder, wow, imagine if in the way we invest in biospecimen banks, each institute invested in digital biobanks in a way where entering a research institution, you could query, what experimental models do I have that have knocked down gene X? And what are the associated phenotyping data? Imagine how powerful it would be for researchers to access such data at an institutional level, let alone at a national or global level.
That resonates, I think, very strongly. And I think that there's a number of missed opportunities with that can be achieved if we can actually harmonize, right? I mean, so people, different entities want to ingest this data at a different level, right? I mean, between the raw data so they can process with their custom approaches, even though we are, beginning to achieve, I think, standardization, at least for certain types of calls, maybe mutation calls, some of the other variants that we like to discover in cancer are those tools are still, I think, improving with time. But sort of this idea that there's a harmonized data set and that we're not sort of losing time in recomputing on these massive data sets. And then there's the opportunity to learn going forward, because obviously it's certainly the case, and I'm sure we've all experienced it, where you layer on another couple thousand samples and lo and behold, the signals there associated with that phenotype that wasn't observable before. And so I think as we really talk about bringing this forward for many oncology patients, not just for those with the most common types of alterations that we perhaps already know how to drug. How do we think about mining those signals? And so there are examples. There's an example in the pediatric oncology space that's been motivated in part because there's just, fortunately, relatively few such patients, and we need to aggregate information and try to sort of jointly learn from it. But I think that we could do better kind of across the board with this. And that also goes to say that, as Elli mentioned, there's these silos. There's silos around clinical trials that have very rich information linked to potentially, at this point, for some of them, long-term outcomes. And I think this outcome phenotypic data that we've talked about is, again, a key element of the data that we need to learn to structure and mine so that we're not, so that we can ultimately do those in silico trials, right? So that we can learn from older chemotherapeutic agents that have been deployed, and now we know genomic determinants of whether a patient's likely to respond. And that's still, maybe not as sexy as our current, targeted therapies, but that's an important and crucial part of precision medicine that I think can be enabled by these kinds of efforts.
Yeah, great examples, Christina. And, thinking outside of oncology in pediatric rare disease, there's been a tremendous willingness to share data. Right. I mean, these are cases where there may, some of these diseases may only occur in 1 in a million or 1 in 10 million individuals. There might be a half dozen cases in the world. And as soon as you can find 2 or 3 of those, it really helps understand how the phenotype relates to a variant that you just can't do as an N of 1. So even an N of 3 gives you tremendous power to get those insights. And we have some great examples of those leading to even treatments that have really helped those children. So clearly that's, at some level the case in oncology too. So I think a good challenge for us going forward.
Patient samples are finite, but once we convert those samples to as comprehensive as possible data, then there will always be development of new methods and technologies that help us to process new signals and new insights from the data. So in some ways, that enable us to immortalize those samples and could be a motivation to seek as comprehensive molecular characterization, be it genomic, epigenomic, transcriptomic, you name it, because there's not just a single use of those data sets, but they will be there forever for the community to mine and reanalyze and aggregate.
Yeah, great point. And anybody else want to add to that?
Yeah, I would just say, in addition to this thought of patients being sort of immortalizing their data, obviously there's additional efforts that involve entailing models, generating models from their biospecimens, including organoids, patient-derived models that, this is becoming more and more reality where there's investigators and companies seeking to do drug testing in these models. And so I think that's an additional source that, again, can enable, to some extent, some of this longitudinal and additional measurements that we might want to make in terms of exploring different therapeutic modalities.
We can immortalize patients through data, and I think that was an excellent point. The sort of in silico immortalization is super important. And in that way, we want to generate as much data as possible from that sample, but also through organoids. We do have a large organoid facility at Tempus. We try to grow as many organoids from patients that we test and store those and allow us to grow those organoids robustly and to evaluate those in the future, biologically interrogate them. But in the organoid space too, just like we need smart tests, we need smart organoids, meaning we have a lot of organoids. Having those organoids connected again to the patient-specific clinical data, clinical history, if that patient's still alive. Hopefully, I think that would obviously, in the best possible scenario, getting continual updates of that clinical and phenotype data, such that the value of that organoid is increased because of the deep, not only molecular, but clinical context around that organoid.
Sure, yeah, and I was even just thinking about in the future, right, in terms of these learnings, how do you go back and as an experimental model then have a system to sort of test this, that you actually knew the genotype and phenotype of the original donor. And I think, it's interesting too, because it also brings the point of, many of the patients that agree to participate in those kinds of programs or seek them out, in fact, are those that really want to be interested, that are engaged in their care. And I think that that's sort of a broader perspective here that we haven't quite touched on, but that I do feel there's, going forward as these data sets are harmonized, as they're democratized, how do we really, partner with the patient in terms of the discovery effort as opposed to exclusively as subjects of research. And I guess I think about this a lot in the context of clinical trials, but even more generally, as the patient, as being the holder of their data and, enabling some of the insights and curation that's possible to really move this forward. And I do think there are trends towards that, and that we'll start to see more and more participation in that way.
I mean, two great concepts picked up there in that discussion. Yeah, one is there's now IT infrastructure that allows people to share their data, right? So, for example, if they have data from, a PopGen study, and with certain providers, through an app, they can then enable others to get access to that data. So, and the interoperability systems and standards are really coming into play. So, we can imagine a day in the future where, if you're an oncology patient and you have your data at Stanford, for example, right, and you want to share it with researchers at Memorial Sloan Kettering, you don't have to go through any gatekeepers to do that. It's something that the individual could decide to do. And that would be a great way for them to give back, potential benefits for them. So I'm glad we're building that infrastructure and hopefully we can do it faster. I also love the idea of immortalizing the sample, because if we think about the kinds of molecular assays we have today versus 5 years ago, right? They're so much richer, more advanced, more sensitive, more comprehensive.
We can think about it beyond research, okay? Right now we're doing, for a practical perspective, in oncology, we're predominantly focused on panel testing, but whole exome and whole genome is not going to be far off. So you imagine you may have been profiled using a more comprehensive assay for a cancer diagnosis, but later in life, one develops an autoimmune disorder or a disease that is being less characterized and less understood. The power that could be in immediately transferring that data for clinical use, particularly the germline data, right? To make new discoveries and on different aspects of disease biology, but also extract relevant biomarkers for the treatment of those patients for alternative causes. That can be incredibly powerful.
So we've talked quite a bit about collaboration around data, really rich topic, so much to do there. Wanted to also see if the panel had thoughts on other areas we could collaborate or the community could, broadly speaking, or where there needs to be, how do we make the sum of all these efforts, more impactful?
I think it was Christina that brought up, I think, that the siloing, which was an important point, which is the siloing of clinical trial data. So I think we're aggregating now the clinical data, obviously, from of course, the normal clinical care at Tempus, which is the real world data is very interesting, and there's a lot we can do with that real world data. But the clinical trial data, I think aggregating that among whoever the trial sponsors are would be very powerful, because there's for a number of opportunities, which for example, the opportunity to rescue failed trials, because the computational tools being developed by folks, in this panel have revealed, well, this failed trial actually wasn't a failed trial because you didn't look at the DNA and the RNA-seq in the right way, and there's this subtype that actually has a fantastic response to this drug, and there's a number of examples of that actually we know that have been published, where post hoc subsets have been identified from a quote-unquote failed trial, but in fact there was actually a very strong responding subgroup that the initial trial didn't find.
Yeah, I think that's an excellent point, and I think there's increased interest in this and appreciation, in part just because the field has moved, right, and this has become so much more commonplace. And I think that this is the time to really be thinking about how to partner effectively in that way, so that we can learn the most from these incredibly, incredibly expensive trials, right? And whether it's biospecimens that were banked, or in some cases minimal data that was collected but really not analyzed using state-of-the-art tools or tools specific for that disease type, as we talked about earlier, that we really need to have that in mind with the context of the phenotypes and what we're looking at in the course of clinical care. It's also the case that the therapies evolve and that we have new tools that might be appropriate, right, based on these learnings. So I really do think the time is very ripe for this, and how to execute on that is complicated when you have so many stakeholders. That's absolutely true. But I will say that certainly, I think that there are the seeds of this moving forward, and that the entire community stands to learn a ton, and the patients, right? The patients who participated in these trials, while they may not benefit directly from those insights, I think that there's a really critical degree of altruism in general amongst the oncology community, patients that are participating in studies and wanting to really pay it forward in terms of those discoveries. So to me, this is just a great opportunity to really bring the fruits of those efforts to bear even further.
So really enjoyable session with my peers and colleagues today. I think really engaged and explored a lot of exciting topics in oncology and how genomics is really moving it forward. So want to thank you all again for your time today and look forward to future conversations.